This is Kevin Mckernan, and I'm obsessed with genomics. And lately, it's been cannabis genomics, at least since probably 9 years now, since 2011.
Hey everybody, this is Jason Wilson with the Curious About Cannabis podcast. Thanks so much for tuning in once again. Today I'm really stoked to have on a guest I've been sort of casually following for years and have been wanting to connect with for a long time. I'm with Kevin Mckernan with Medicinal Genomics. Thanks so much, Kevin, for being willing to come on the podcast.
Thanks for having me, Jason. I've really enjoyed all the guests you've had so far. So it's a great podcast you got going.
Oh, thank you so much. I appreciate that. And I look forward to having you right in there with everyone else that I've been speaking with. And today we're going to talk about a topic that I haven't touched in any other interview, primarily because it gets into an area where there aren't a ton of people doing this work, but we're going to talk about cannabis genetics and genomics and microbiology primarily. And so, Kevin, for those listeners that maybe are not familiar with you and your work, can you briefly describe some of your academic and professional background leading up into your founding of Medicinal Genomics and the work that you're doing there?
Oh, sure. So I started off my genomics career at the Whitehead Institute back at MIT. So this is a center in Cambridge that was sequencing the human genome and was part of an international effort to sequence the human genome along with the Sanger Center and a few folks in Japan and Europe. That was from 96 to 2000, and I was managing their research and development team there. So we built a lot of the robotics that automated the DNA purification and got the sequencing running there. And in 2000, when that project was coming to a close, I decided to spin out a few of the patents that we had acquired at the time there into a company called Agincord. And this was a private entity that was a DNA sequencing provider for a lot of people in the pharmaceutical and healthcare space. But we also sold DNA purification tools that help people extract DNA out of cells. And fast forward to 2011, 7 years in that time frame, the company also built a DNA sequencer known as a solid sequencer that eventually went to market under Applied Biosystems. They were for those in the sequencing field, they probably know of maybe two companies that have had fairly large market shares throughout the history of DNA sequencing, and ABI was the first one. They had all these Sanger sequencers, and they have they're still in the market and they still do their thing under the Thermo name. But there's now Illumina, who's a very large contender and arguably a larger entity now. And we were competing with Illumina with the solid sequencer for a period of about 5 years. And then in around 2011, I decided to just do something different and get out of the industry and go into the plant genetic space. And we began sequencing cannabis genomes back in 2011. Flew over to Amsterdam and extracted a bunch of samples over there, met Don and Aaron from DNA Genetics and Dave Watson and a bunch of other, Robert Clark, a bunch of other folks showed me around over there. And we really didn't know what to do with it at the time. We just knew that genomics helped so many other markets. It was bound to help this one, but we had no idea what to do with the information and how to turn it into a company. So we kind of explored a little bit. And I'd say it was a very slow start with medicinal genomics because we were a company that was embedded inside of a clinical diagnostic company known as Cortigen. Mm-hmm. And most of the company's efforts were sequencing kids with epilepsy and mitochondrial disease, and arguably a lot of diseases that center around the ECS. Although the people in the clinical sequencing space probably don't know what the ECS is yet. Right. They're, as you've probably heard from many of the guests you've had before, it's not really taught in medical school. So that was an interesting business, but a very challenging one because as insurance companies moved into the space after the AMA, they didn't like this idea of sequencing for idiosyncratic diseases, diseases we don't really understand. Yeah. They don't want to reimburse for that. And so they started raising people's deductibles and making that type of sequencing very difficult for people to do. And that's when we decided to shift the company fully into sequencing cannabis genomes because we saw, ironically, less headwinds, regulatory headwinds in the cannabis space than in the clinical sequencing space. So if that gives you any sense of the background, I've wandered from research sequencing to clinical sequencing all the way into what people might call recreational sequencing, but always has DNA in the thread.
And what is it about working with genetics and studying DNA that really draws you in, that you find really interesting?
Well, Personally, I like the fact that it reveals how different everybody really is. I mean, we have this societal tendency to want to group people into either races or sexes or these other large categories and treat the entire population as one. And what we're learning from the Human Genome Project is that is incredibly stupid. Everybody's very, very different and unique, and personalized medicine is clearly the way to go. And it's just taking time to sort of fight the system, to make the system aware of the fact that you're not going to have an algorithm to apply to a population. You're going to need to have physicians actually speak to patients and ascertain that individual's needs. So that, I don't know, that has always been appealing to me, as I generally don't like one size fits all principles. They're just grotesque, and there's a tremendous amount of potential in sequencing people's genomes and improving healthcare. Now that same story really resonates in cannabis, because you get involved in cannabis genomes and you realize they're even more different than people are. And this concept of a sativa or a hybrid or an indica is garbage.
Yes, I'm glad to hear you say that.
And so we have to change that, particularly if we want the medical system to start to speak this language. They're not going to prescribe green crack to somebody. It's they're going to want to know terpene profiles, maybe even a genotype, because there's a lot of dark pharmacology, as you heard spoken about before. The chemotypes are great, but they only measure maybe a dozen compounds, and we know there's thousands. And so the genomics is another cheap way of getting at that information. And I think the other thing that's really exciting about the genomics field is there probably isn't a field out there that's moving at a faster pace. When you start looking at the cost declines in DNA sequencing, we've never seen anything like this before. And even in the chip market, where you hear about Moore's law, and Moore's law is slow. It's actually something that kind of gets laughed at in the sequencing space, because we're always held back by the fact that the chips aren't fast enough. And the sequencers are moving at a faster pace than the semiconductor improvements are. So it's one of those fields that forces you to learn about DNA storage and data storage information, whether that be in DNA or not in DNA storage. You have to learn about hard drives. You have to learn about internet bandwidth. You have to learn about chemistry required to sequence these things. So it's very, you can't get bored in this space.
Yeah. Man, there's several things you've touched on that like spark passions inside of me as well. I mean, one, like I mentioned before we started the interview, part of my background is in IT, and even in, chemistry labs that I've worked in, a lot of times I end up becoming the technologist, because I'm very interested in the equipment as well, and the idea of information. And so one thing about genetics that interests me, but I'm so deficient in my like formal training with genetics, but just the idea of how information is stored, transmitted, in these ways that are not necessarily intuitive. But then also what you just touched on is something that several clinicians that I've interviewed have talked about, is this move towards what's sometimes called N of one medicine, or individualized medicine, trying to figure out how to use that genetic data to understand what a patient is sort of bringing to the table before going in to prescribe certain treatments and everything, so that you can narrow down with much more confidence what might be more likely to work.
Yeah, and I think it's really important. We learned this in the epilepsy field, that sequencing and understanding the patient's genome can save lives. I mean, there are certain mutations in epilepsy that just require vitamin supplementation, and the seizures go away. And that can often get in the way. You might put them in CBD and not notice that for 6 months, and then they end up honeymooning out of it, and it really turns out they just actually needed vitamin B6 or B12 or folic acid, or they need to be on a ketogenic diet because they have glucose transporting mutations. There's a host of, there's at least like 24 different forms of genetic epilepsy that are actionable based on the genome itself. And they may not always lead you into benzos or cannabinoids. They might lead you into a path that doesn't even involve these compounds. So we think it's very, very valuable. The challenge is cannabinoids right now sit in this really weird legal conundrum where it's difficult for doctors to prescribe them. It's even more difficult for you to have a genetic test that actually tries to look at the variants and make informed decisions on which cannabinoids to potentially use, or if to use them at all, because there's a lot of there's a lot of FDA scrutiny on direct-to-consumer genetics.
Right.
There shouldn't be, but there is. They don't like having patients enabled to make intelligent decisions. They want that all to be channeled through the FDA-appointed medical school doctors, and that doesn't scale. It's important that physicians obviously are very well trained and know what they're doing, but the better thing is to make sure all the patients begin to pay very close attention to their health, because there's no one who is more vested in the outcome of their health than the individual. And arming them with information, in my mind, is always a good thing. But they don't see it that way yet. And so they have been cracking down on a lot of companies that do direct-to-consumer genetics if they ever imply that they're going to be used for medical decisions. 23andMe was one case of this, where they just got hammered by the FDA, and they kind of sent a signal through the field that if you do any of this without going through the FDA, we're going to come down on you. So we for a while, we're looking at panels that would genotype patients and try to discern, whether they had like an MTHR mutation and they should go one direction, or whether they had variants that might predispose them to opiate addiction, because there are some known that increase your likelihood of opiate addiction, and that it might be that might coach you in the direction of cannabinoids. But I think the sequencing needs to get a lot cheaper for that to happen, because right now it's probably a couple $100 might be better spent going to a dispensary and just experimenting with different CBD and THC ratios until you find something that actually works, because the science we have around the genotypes is still, I wouldn't say it's fuzzy, but it's early. It's very early correlating these genotypes to outcomes.
Well, that's exactly what where my mind was going next. I know that one of the limitations currently with some of the genomic research on humans is just trying to understand how to interpret the data that's there and how to correlate it to very specific things. And something that I think we'll get into in a little bit when we talk about cannabis is that genetics can be very tricky in that you can think you have the full picture of what genes and what correlating enzymes and things are responsible for trickling out into certain effects, and then you realize the picture is more complicated than you initially thought, and there's more involved, more genes, or even novel mechanisms that you weren't even aware of.
It's very true. It's an onion, and as you dig into the genetics, you realize there's many more layers to the data, from the transcriptomics, the proteomics, to all the methylation that's going on. So it is it can in fact be a rabbit hole in that regard. But on the bright side, this is a plant that's largely not been studied yet. And when you look back over the last 20 years where they have applied genetics to plants, the productivity gains are just massive. The agricultural revolution that's happened is clear as day now. That since they sequenced the rice genome and the wheat genome, you can see the number of traits that they're starting to discover and how they can use that to breed for more efficiency. It's something when you see those papers, you start to recognize that like the carrying capacity of the planet isn't as limited as we might think.
Yes,
that we are going to get more and more effective at biology. And how we as humans fit into that biology in a way that isn't as wasteful as it is today.
Yeah, exactly. I mean, something that comes to mind is like, in the classic story and movie Jurassic Park, where it had the idea that life finds a way. Studying these things helps you understand how life is so adaptive and able to, reveal different traits that you never would have imagined might have been possible in order to, continue persisting throughout time. And it's interesting to see humans' role in that. I think something that I think about a lot, sometimes people have a tendency to talk about humans as if they're outside of nature.
Right. Yeah.
And in the sense of this work, it's, I don't think of it that way at all. Like we are part of nature. We are part of how life is unfolding and interacting with itself. And so in this funny way, people studying genomics, doing this work, unlocking these tools and understanding how to be more conscious in how we influence life and that sort of thing. It's interesting. Right. It's like Mother Nature doing these things, through itself, but using humans as a tool to this meaning.
It does bring up a lot of controversy, right, in terms of GMOs, and are we part of nature when we start modifying nature, and is this analogous to beavers building dams and bees building beehives and all the other tool builders out there? Are we meant to utilize our intellect to do this to nature? And that's a deep topic that can go on for days. But my general sense on it is there's multiple different ways one can be performing GMO. I mean, arguably Mendel was doing GMO. He was genetically directing a breeding program. And we can do that today too, but even at a faster pace because we have basically better measurements. We're not just looking at maybe the color of the peas. We're actually looking at the cannabinoids, which you can't see with your eyes, or you're looking at the genes that encode them so that you can track these things at a much faster pace. But I think we're also going to be confronted in the next few years with how does the community feel about removing genes from the genome? Not actually adding any foreign content, but maybe it's important that we delete a couple rare cannabinoids so we can comply with the USDA's hemp laws. Right. There are some leaky cannabinoid synthase genes in the genome, and some of them you can get rid of, some of them you can't, depending on what your goals are. And I've seen rumors and maybe even a couple patent applications implying some people have gone about deleting at least THC synthase. I don't think you need GMO to do that because you can find plenty of plants that don't have it and just breed for it. But let's say it was some other gene you wanted to get rid of. I think that's going to come our way. I think the other thing we're going to see are people who are trying to be very cognizant of the fact that GMO has a marketing problem right now, that they're going to try to do manipulations with the plant's own genes. They're going to maybe take the chloroplast genome and add stuff to it because the chloroplast genome sits at extraordinarily high copy number, and you might get more expression putting certain genes there. There's other people who have thought about putting other markers on the Y chromosome as well. So you can find males from a drone flight. And
interesting.
That's probably going to require a foreign gene. I don't know of a gene in cannabis offhand that might, in fact, produce that marker. But the obvious one is green fluorescent protein, which was pulled out of a jellyfish. That's used to mark all types of other transgenic plants, and you could pop that into the Y chromosome and have your plants glow in a certain wavelength such that a drone flight could identify males or rip them out. And those males presumably won't propagate if you're good at removing them. But there's always the risk that they'll make pollen that could spread. And then there's a property rights issue as to who, did you get your pollen into my field and who's responsible for that? And with all of these modifications, that also tends to bring the elephant into the room, which is patent rights. It's easier to get patent rights on things that are clearly man-modified than things that have been naturally bred. There are breeders' rights, there are plant patents, and there are utility patents, which we're seeing roll out right now, and they can cover naturally occurring plants, but it's far easier to get something through the patent office if you've clearly changed it from nature. I don't necessarily agree with that moral philosophy, but that's the facts of the matter. That's what we have. So I think we're going to be faced with a lot of these things sooner than we realize. If you actually go through the patent office today, you will find patents in there that have cannabis in there where they have put glyphosate resistance into them. They have put human P450s into them to modify cannabinoids so that they are more water soluble upon extraction. There have been ones that I suspect they're going to be ones working on C3 to C4 photosynthesis conversions. And there's a host of these mods that are already in the patent office. This doesn't mean the technology works yet. Right. Patents have no obligation to work. There's anti-gravity devices out there, right? They're just ideas. But usually people don't file those unless there's some preliminary data because the expense of filing isn't something you just do on a brain fart. You actually want to make sure it's worth calling the lawyers for. So I know I see a wave of all of this innovation coming into a community that has very diverse opinions on it. And I'm kind of curious where that's all going to go.
Yeah, I agree. It'll be over the next 10 to 20 years, it's going to be very interesting to see how everything evolves and how the industry accepts certain technologies and rejects others. I'm very, very interested, especially when you have, like you were talking about just a second ago with THCA, you have these market dynamics influencing some of that, where you've got people that are more so maybe now than ever feeling that pressure of needing to find genetics that are going to ensure that their livelihood stays intact now that the Farm Bill's passed, and we'll see how it changes over the years. But I know that's why there's a big push now for CBGA dominant cultivars and everything, but they're still getting THC in some of those. So it's interesting, and this starts to segue into some of the research I wanted to talk about. But before we get into all of that, to set up a good foundation, can you describe a little bit of some of the tools and techniques that are used to study DNA, and kind of keep in mind that some of our listeners don't have any background with any of this. So,
sure.
Yeah.
Yeah, absolutely. So there's generally two different types of DNA sequencers that are out there. There are sequencers that are used to resequence a genome, generally at much lower cost, if a reference genome is already known.
Gotcha.
So let's say like we did the human genome back in 2000. It wasn't really finished and still isn't finished today. There's still some centromeres that haven't been fully sequenced yet, but 99% of the genome's known. There's a large effort trying to make sure we have maps across all the telomeres and centromeres right now, and they've managed to traverse a couple chromosomes from telomere to telomere. But once you have that reference genome, since the human population is fairly inbred, we differ maybe by 1 in 1,000 letters in the genome. What that means is you can use a much cheaper sequencer to resequence other people's genomes and then just compare it to the reference. And this is usually where Illumina comes into play. They can only sequence about 100 bases of DNA, maybe out to 300. In some extreme cases, they can do paired ends or 600 base pair reads, where they do 2 300 base pair reads aimed at each other. And that gives you, it does really well in the human genome. You can remap and sequence people's genomes for probably under $500 now. That doesn't include all the analysis, but just the raw cost of the sequence is getting close to that point. Now, with that said, when you're resequencing and you're mapping to a reference that is fairly representative of the entire human population, you can figure out where all the single letter changes are really easily. It's a bit more challenging to figure out where entire chapters of the genome are missing with those technologies, because when the read gets shorter than the size of the variant, it's very difficult for those reads to actually detect the variant. So as you approach like 25 to 50 base pair changes in the genome, the short reads systems start to stumble and miss important genetic variation. So there's been an effort to increase those read lengths, and most of those efforts have gone in the direction of sequencing single molecules of DNA so that they get a cleaner signal. The Illumina systems right now, in order to read the DNA, they first amplify the DNA to a couple thousand copies, and then when you're sequencing that particular colony, you're actually sequencing 1,000 molecules that are all marching in step down the DNA strand. And what invariably happens as you go out in read length, each one of those reactions is probably 99.9% efficient, but over time, they start to get out of phase. The molecules get out of sync, and it gets very difficult to start reading the sequence. So to get around that synchrony problem, what the direction the technology moved was to sequence single molecules, which is much harder to do because you need to be 1,000 times more sensitive. But once you're reading single molecules, you never have to worry about the rate at which the polymerase decides to read the molecule. You just let the thing run, and you get to read the DNA as the thing is copying it in real time. So the two sequencers that are probably most known for this are Pacific Biosciences sequencers, and then an upstart sequencer that's I probably have one floating around here somewhere. It's called Oxford Nanopore, and they're much smaller footprints, but they're much noisier. These things will thread DNA through a biological protein pore, and when the DNA gets threaded through that pore, the current starts to change on you based on the sequence context that's in the pore. So the thing might zip through at 500 bases a second, and as it's doing so, it's leaving a current profile that tells you some information about the sequence. They're about 90% accurate. They're getting better. It's a very quickly improving platform. But the interesting thing about that platform is it's so small, it's portable. You can bring it to coronavirus outbreaks. You can bring it to Africa.
I see. Yeah.
And then you usually need a hell of a lot more bioinformatics in the back end to clean up the signal. the read length on those are unlimited right now. They have had reads go out to 2 million bases on those things. So it's a massive breakthrough on read length.
Wow.
Somewhere that's a little bit more of a compromise between the two is a system that also sequences single molecules, but the way it reads it is using a zero mode waveguide, which is basically an array of very, very small holes. And there is a phenomenon when you're trying to image molecules, single molecules in particular, which are very small, is that the problem you have with anything that uses light or fluorescence is that fluorescence is actually a fairly large wavelength. If you look at the wavelength of blue light, for instance, is 500 nanometers, and you're trying to measure things that are angstrom. So it's kind of like shining a floodlight on top of a speck of sand, trying to watch the speck of sand. And if you have more than one speck of sand in the floodlight, you really don't know what you're watching. And so what they had to do is build these tools that focus light into a smaller diameter than the actual wavelength of light. And that's done with a technology you can see on the front of everybody's microwave oven. A microwave oven actually has this zero mode waveguide there that prevents those much larger wavelengths from getting through. But what happens at the surface of that, never put your hand directly on the surface of that thing. It won't let you because there's a screen there. But if you were to get close enough to that, there would be a zeptoliter volume of irradiation coming through there. And that zeptoliter is enough to illuminate a single polymerase molecule copying a DNA strand. So PacBio does this really elegant device where they read 8 million of these holes at a single time. And they've managed to be able to put circular molecules into this thing. So when the polymerase grabs onto it and rolls and starts to copy it, it runs it around like a gerbil wheel over and over and over again. So you end up reading the forward strand, then the reverse strand, then the forward strand. And as you do this 30 or 40 times, you eliminate all the noise. And they now have 20,000 base pair reads that are 99.9% accurate, which is like an absolute remarkable breakthrough. And we've only had that in the last 4 months.
Wow.
So much of the work that you see in the paper we put out actually used a technology of theirs that was a little bit older, that was probably 50-fold less accurate, but it had 200,000 base pair reads. We use that to assemble a few of these cannabis genomes. And very recently, we've gone back and resequenced them with this newer hi-fi chemistry that they have to improve the references even further. But it's those two main tools that I think are being used the most in cannabis right now, is that we don't have good references, and the human genome is easy in comparison to the cannabis genome to sequence. A couple of the things that make genomes easier to sequence is that if the mother and the father genomes are fairly similar, they're easier to sequence. And that's the case with humans. In the case of cannabis, there's probably a variant every 50 letters instead of a variant every 1,000 letters. So that's much more difficult to sequence because the mother and the father genomes are like sequencing two different genomes that are blended together. And that can confuse a lot of the algorithms trying to put the sequence back together. So the long reads on these platforms, and particularly the long ones that have accuracy, are what are really shining through. They're the ones that are putting the genomes together in perfect order, near perfect order right now. And that's enabling us to start to ask, can we build a single reference for cannabis where we can resort to these cheaper tools for mapping reads? And I think the verdict still out on that. We do a lot of Illumina sequencing. The preprint that we have up there has 40 genomes that were done with Illumina mapped back to the reads that were the references that were built with PacBio. But one thing that we discovered doing this is that while there's a single letter change every 50 bases, we're finding there's an equally high rate of entire chapters being deleted. This is what people would call structural variation. And so that preprint listed about 58 million single base pair changes in the 40 genomes that we looked at. But in one of the trios that we sequenced, which is a mother and a father and its offspring, we did that all with PacBio, so we had the visibility to see these large chapters being deleted and added. And we're seeing upwards of about 1/8 of the genome that's in structural variation between just an inbred cross. That's an enormous amount of genetic material that is different just by crossing siblings.
Right.
So let alone if you step out of the cross like a type 1 with a type 3, it's probably going to be even more diverse than that. And it's beginning to beg the question whether we should go with the old route like we did with human, where we make a reference genome and map everything to it. It might just be the time and where the technology is mature enough where you just forget the short reads altogether and just always use something like PacBio. That's a cost question. Right now, PacBio is probably tenfold more expensive than Illumina, so a lot of people want to try and leverage the Illumina platforms as best they can. But if you really want to know what's going on, a single sequel chip from PacBio, which are usually a couple grand, you can probably get a trio sequenced on one of those now. So we tend to recommend people in breeding programs, if you don't know where your parents came from and you're not confident in them, at least sequence the parents with PacBio. And then you can do all your F1s and F2s with the cheaper tools. But if you're starting with a distant reference, like the ones in NCBI, where you don't even have access to the germplasm, and they may not even be the same type of plant. Most of the references up there are type 3 plants. You're going to have a bad time. There's so much difference between those references that they're a great starting point, let's say. But I think the direction the field's going to go is in these toward a pan genome, where if you're going to build a reference, it needs to include type 1s, type 2s, type 3s, type 4 plants, so that we have all the industrial uses of cannabis in there as a reference, so we're not missing genes.
Right, not just focusing on the drug type cultivars and.
Right.
And
or the hemp one.
Right. Yeah, exactly. Yeah. And can you describe really quickly for our listeners, what are type 1, 2, 3, and 4 cannabis varieties?
So this goes back to Etienne Demeyer's work and probably Dave Watson's and a few others. They classified cannabis into 5 categories. These need to be refined, but this is just a historical classification where type 1 plants are THC dominant and tend to have a 23-to-1 ratio of THC to CBD. Type 3 plants are CBD dominant plants and have the inverse relationship. Type 2s have both genes and they make both THC and CBD. And as you get into type 4s, they don't have either gene. They don't have THC synthase or CBD synthase, and they just make the precursor cannabigerol. Type 5s are broken even further upstream and don't even make cannabigerol. And so they're cannabinoid-free plants. Now, what we've come to learn after doing all the sequencing is that was a remarkable classification system they figured out, not having ever seen the genome. They figured all this out kind of the old school way with Mendel. So they don't really know, or never published where those markers are in the genome. The preprint we put out at least attempted to do the most popular ones that we found. And the most common variations we see in these cannabis genomes that map to this nomenclature system are the type 1 plants have a broken CBD gene, so they really only have THC synthase there. And whatever CBD is coming through is probably a promiscuous synthesis from the THC synthase gene. These genes are not 100% pure. They don't make only one compound. They tend to predominantly make one compound, but leak a little bit of the others. Likewise, if you look at the CBD plants, they usually have the THC gene completely deleted. And the reason why we think the nomenclature system probably needs some more refinement is that there are some rare cases where you can have a THC gene be present in a type 3 plant, but it has a point mutation in the gene that's deactivating it.
Gotcha.
But the gene's still there. And so that can throw off some testing tools. We've had testing tools in the marketplace now for 3 or 4 years that try to just look for the popular ones, the full gene deletions, but they get tripped up when there's actually a THC gene there and there's a point mutation that's killing it because the assay is not sensitive to pick up that single base change. Likewise, if both of these genes are missing from the genome, it's usually a good sign that it's a type 4 plant. And so a lot of people have been using the sequence information to breed for CBG plants right now, because when you have both THC and CBD synthases deleted, you have less synthase genes that could be leaking THC expression. And so the other genes that we're paying close attention to in that preprint are the cannabichromine genes, because those are the next closest related genes to THC synthase. And the plant, for some reasons, made like 8 of them. It has got more of those genes than the THC and CBD synthase genes. They're not heavily expressed in a lot of the tissues we've looked at, but again, we haven't looked very deep at I think to really find cannabichromine expression, you might need to be looking at different time points in flowering and maybe in veg states to really see it turn on. We happen to see the most cannabichromine expression in male plants, and I don't know why.
Oh, interesting.
That was the only place we actually got RNA expression in that study, were in male flowers. the Jamaican lion plants that we sequenced have a cannabichromene cassette. A lot of plants don't even have that cassette. So, but that's another gene that people like to keep their eye on. If they're trying to breed a wider spread between a cannabinoid and THC that's compliant with the USDA regs, is they pay very close attention to the copy number of THC synthase, CBD synthase, and CBC synthase, and that usually gives you some guidelines as to how much you might leak.
Yeah. Have you, going specifically to CBC just for a second, in any of the work that you've done, have you seen any correlations with differences in environmental qualities that might influence CBC in any sort of way? And the reason I ask is there's some old literature. I mean, possibly, I can't remember when it came out. It was possibly late 90s, early 2000s, so it could be totally irrelevant now. But it was based on looking at some of the data that was coming out of the University of Mississippi, and some of the accessions that they had, they were seeing higher levels of CBC, and there was speculation that it might have something to do with humidity or something like that, given that the plants were grown in Mississippi versus more arid environments where they normally would be grown.
So it's a really good point. And one thing that surprised us in this whole study is after we sequenced these 40 genomes, and there's a figure in the paper I should point people to in this that may give them a perspective on it. Let's see which one it is. I think it's figures 7, 8, or 9. What we looked at was the presence of these copy number variations. Your audience probably can't see this, but what shocked us, we'll maybe post it in show notes or something, is that the blue means they're deleted and red means that you have the genes. The Jamaican lion strains are here purple. They're heterozygous for it. But what was shocking to us is that there are all of these different strains out there that don't have this cannabichromene cluster. And this isn't one gene. This is 2 billion bases of sequence that's deleted.
Wow.
And what's on that contig that's been deleted, or the portion of the contig that's deleted, happens to be 3 or 4 other genes that are involved in pathogen response. There's an aquaporin gene in there. There's a few of them listed in the paper, but so if there's any cannabinoid that would be most directly linked to environmental conditions and perhaps pathogen response or humidity, I would pin it on that one, because you can see that there's a common deletion in the population that when it's gone, you lose some of these other genes with those cannabinoid synthase genes. And so those plants arguably are either going to be more prone to pathogens or have a different response to stress one of them is actually a gibberellin transporter, right? That gibberellin's a really important plant hormone, and I don't know how many more the genome has. I have to go and look at that, but just seeing one of those things get deleted means the plant's probably going to have a really odd response to stress, and it's in very close proximity to the cannabichromene genes that might be reason to believe that the cannabichromene expression might be a little bit more linked to these types of changes.
Wow, really fascinating. This is already like making the gerbil wheels in my head spin, because that's something I've always wondered about. And hearing about this genetic work is nice, because with my background looking at the chemistry of the plant and doing testing, that sort of thing, CBC is one of these problematic cannabinoids where if your method is not really dialed in to really measure CBC accurately, you can get a lot of other compounds that can collude around the same time as CBC. They're isomers of other cannabinoids that show up around the same time. So, something I say to a lot of cultivators and extractors that are trying to get information on CBC is, some of the data that's floating around on your test results about CBC, you can kind of take with a grain of salt. That might be CBC, might be something else. It's one of it's just coming out of the labs. I just know it's one of those problematic cannabinoids that a lot of times is not super accurate compared to, CBD, THC, or CBG even.
Well, that's an industry-wide problem in that we have a hard time as it is correlating some of these genetics to the particular chemotypes, because we're usually relying on genetics from all over the world, but then chemotype testing that's done in 50 different laboratories that all run different methods, and it's very hard to standardize and get results that you believe in unless you take control over all of that and run it locally. And
exactly.
That means you're usually studying plants that are only within your state, because you can't get the flower material across state lines legally unless you're dealing with type 3 plants. So there's a lot of limitations, I think, to the research because of that. And as your other casts have mentioned, we do need to be looking at the flavonoids, canflavin A, B, and a variety of these other cannabinoids that people are starting to find, like the Italians. The THCP cannabinoid is probably at very low levels in some cultivars that we just don't know about because no one's been looking for it and there aren't standards for it yet.
Exactly. Yeah. I mean, the cannabis as a biochemical factory is, the potential there is really incredible. It's, something I also say to people a lot is, if you're a researcher wanting to get into cannabis research, it's an exciting time because there's, depending on, if you can think outside of the box of just THC and CBD, there's so much low-hanging fruit to pursue regarding research projects and stuff. So there's so much work that needs to be done. And that's why it's exciting. I have mixed feelings about the way the hemp industry is going. But one thing that's exciting with labs needing to get DEA licenses to handle hemp, this changes the game as far as labs being able to work with like universities or even other labs that have been kind of slowly trying to do cannabis research on a federally legal level and have kind of struggled, and there hasn't been a strong network of researchers there to kind of support each other. That's going to change a bit, and like already I've done a little bit of work with some universities to help them get sort of caught up on some of the scientific literature and get some analytical methods in sort of development and moving towards validation and stuff. And I'm really excited to see what momentum we're going to see when these labs are able to actually move material to one another, and universities are feeling comfortable not risking, their funding and that sort of thing to actually touch cannabis.
And that is starting to happen. We're seeing Cornell get funding now. We're seeing Harvard get funding now. But you're right about the DA issue. That's almost going to guarantee that labs that know the least about the plant are the ones that are put in charge of it.
Yeah, it's a good point.
And the testament to this is just look at what happened to Full Spectrum Labs, right? They were an operating cannabis lab who tried to play by the book, applied for a DEA license, and since they were at the time presently handling cannabis, they got raided and shut down. Right. So anyone who's currently right now handling cannabis, they're going to be really reserved to go and apply for a DEA license, knowing what's happened in the past. That's almost going to invite a DEA raid, unless you set up in a completely new business that's financially untethered and then somehow get your intelligence in there and then apply. It's just it's certainly going to favor pre-existing DEA laboratories. And from the ones that I know, they're great at picking up cannabinoids in urine, but they're awful at picking up the plant.
Yeah, they've been focused on toxicology, and that's really the only thing they've been really working on. Yeah, it's going to be interesting. There's going to be a learning curve for sure that we're going to have to wade through over the next several years while labs get caught up. But yeah, thinking about the long term, I think it's all. I mean, of course, I'd rather just see cannabis descheduled, and then you don't have this.
Right, and then none of this is
right.
DEA purpose to study a plant is kind of crazy.
It is. It absolutely is, and that's something I always try to preface. Is as much as I talk about, part of my job on the side is helping people navigate these regulatory waters, and so I talk about it a lot, kind of casually, but I do like to point out it's like I don't want it to be this way. I definitely don't. Yeah, but it is, and and it's what we have to work with until it changes. So segueing into the research that we've been sort of dancing around, let's talk about the study that just recently put out. So right now it's in a preprint form, but so there's a ton of data there. In this research, you looked at Jamaican lion cultivar and mapping.
That was. Oh, go ahead.
Oh, I was just to say you were mapping the parents, the siblings, and you were looking at genes related to cannabinoid expression, sex expression, and pathogen resistance. Is that right?
Yes, yeah, and so just to caution all of it, it is a preprint. It's not been through peer review, but all the data is public, so anyone can grab the data and download it and play with it while it's going through peer review. Koji is a great place to go if you want to look at the data. It's a genome browser. Comparative. If you just Google COGE or comparative evolution or comparative genomics genome browser, you'll see a search in there for Jamaican lion or for cannabis, and you'll see a certified reference up there that has a lot of this data linked in. It's got great graphical tools, so you can kind of play around with this data yourself. But yeah, so the purpose of that project was to first build some good cannabis references. Back in 2011, we sequenced a ChemDog strain with these short read Illumina tools. Mm-hmm. And we probably did 1 billion reads. And when you try to assemble those reads, the genome is so complex that you only get like puzzle pieces out that are about 3,000 letters long. This is what we call a contig size in genomics. And you want those contig sizes to ultimately be the full length of a chromosome. Mm-hmm. It's rare they ever end up that way. They usually are broken up into a couple pieces because the centromeres are hard to get through and some of the telomeres are hard to get through. But the human genome has 1 or 2 chromosomes after 15 years of being declared finished or done, finally tip to tip. So we're going to do much better than that in cannabis. It's going to happen faster because we get to stand on their shoulders. But so there's this term in the genomics field that measures the contiguity of genomes, and it's known as the N50 number. This is the average size of the length of the pieces of your puzzle. And cannabis chromosomes are usually 60 to 100 megabases in size, and you'd love for your contigs to be perfect stretches of 60 million bases long. I think the largest one we have in one of ours is 40 megabases, so it's close. We're almost to perfection. But the references we had when we started this project, the N50 size, I think the largest one at that time in GenBank was about 150 kb, so 150,000 base. So a big improvement from 2011. And then John Page's group put a few out a few days after we released the Jamaican Lion one. They put one out from Purple Kush and Finola, and those inched up toward 400. I think Sunrise put one out that's in the 750 range, 750,000 bases. But the real assembly gurus don't get excited until you're over a megabase. That's kind of like a mental threshold that they want to see before they start considering a reference grade genome. And we still weren't quite there yet. So we decided to keep sequencing, and all that we did different than the people behind us was that we just jumped on board of the newer sequencers. The latest and greatest chemistry just pushed the Jamaican Lion reference above the megabase mark. So it's currently at around 3.2 megabases on the preprint that we put forward. So the average stretch of sequence in there is a stretch of 3.5 megabases long. Now, the other thing that's important is looking at the quality of the sequence to make sure there aren't a lot of errors, because when you have 58 million variants, you don't want to be spending your time on 5 million ones that are wrong. It's a lot of error to sift through. So the more later sequencers tended to have a lot better error rates and more accurate data. Now, the other thing that you tend to do to really build a strong reference genome is you like to sequence a trio, which is a cross, basically, a mother, father, and an offspring. And the reason you do that is you can then triage your errors that don't make any Mendelian sense from the ones that should. We call these Mendel errors, and there's less than like a 0.2% Mendel error rate in the Jamaican Lion trio that we have. So what that means is you look for variants in your offspring, and you say, do I see evidence of this in either of the parent? If it's not in either parent, it's probably a sequencing error. And you use that information to basically categorize what variants are real and what are wrong. The other thing that usually has to go on when you're dealing with a new species, and we're doing some work with this on Google and a few other parties in the space, is you usually have to retrain your algorithms that look for the variants when you're dealing with a genome that's as polymorphic as cannabis. All these tools were trained on humans, so they're only expecting a variant every 1,000 letters. When they start seeing one every 50 bases, they start throwing everything out, saying this is a different organism, get the hell out of here. So you have to go back and retrain all the base callers and all the variant callers to start calling the variants more appropriately. And they usually take the tools that are used for mosquitoes. Mosquitoes have a variant like every 20 bases. They're one of the most polymorphic species out there. And so the tools that they did on Gambi, they can then, Anopheles Gambi, they can then kind of tune to be somewhere between humans and Anopheles and settle in on cannabis. So we went through the effort of doing that type of training with the tools that we were using, so we get the base calls really accurate and the assembly is very accurate. And then once you have that sort of trio, that reference set, you can then confidently take Illumina data and map it across all of those references and start asking questions like, are there entire segments of the genome missing? What's the polymorphism rate in all the genes that are out there? How many highly damaging variants do we have in a given cultivar? In other words, mutations that actually destroy genes. Right. How many of them? There's like over 2,000 of those in a given genome. There's that many genes that get obliterated through polymorphisms. Once you also have these references, the next thing you want to do to curate them is you go and sequence the RNA off of as many tissues as you can. And what that does is it tells you what genes are being turned on and where they are in the genome. Today, we don't have great tools that can just scan a reference genome and say, oh, I can see where all the genes are just algorithmically. They really want to see biological evidence of how those RNAs are getting written off the genome. So if the listeners are unfamiliar with DNA and RNA, the analogy you often hear is that DNA is like information on a hard drive. And the RNA is what's in your task manager, which programs are actually getting executed off that hard drive. Not every cell in the genome or in the plant is going to be expressing all genes all the time. Right. A trichome is going to be expressing a lot of THC synthase, but it may not be expressing any of the root proteins that are needed to interact with the mycorrhizal. So you do this to try and get as many pictures of what genes are on and which tissues. This helps you annotate the genome so you know where all the genes are. And I don't think we're done with that process yet. We did this across 5 tissues. We found about 27,000 genes in the female genome and about 32,000 genes in the male genome. And those numbers are likely to increase over time as people survey more tissues and do more sequencing. One thing that is really a bit of a breakthrough that's happened this year in the sequencing platforms is those same single molecule sequencers can now be deployed on RNA molecules. And in the past, we didn't really have that. We had these short read sequencers trying to ascertain where the RNA is mapping to the genome. And that's really hard to do because RNA is kind of like a fragmented hard drive. Mm-hmm. It never reads a gene from a capital letter to a period as a perfect sentence. Now, what tends to happen in the genome is your gene gets scattered across the genome in like 10 different pieces, almost like fragmented data on a hard drive. And there are tools in the cell that when you make that RNA transcript, they go and splice out the regions that are the exons and make what's known as a transcript. And the process of excising those exons, it can do in a combinatorial way. So you can have any given gene through a process known as alternative splicing of that RNA can make 3 to 10 different proteins. So the protein complexity is not a one-to-one correlation with the genes that are in the genome. But having these sequencing reads that can read across the entire gene's RNA in one shot means you can accurately place all of those exons back into the genome. Wow. We haven't had that in cannabis until this year, actually. I think we were the first people to probably put out any PacBio RNA isoseq data that gave us really clean information on intron and exon boundaries. That's something that's really hard to do with short reads, particularly in very polymorphic species. It's a bit of a challenge. So we now have that backbone inside that Koji browser. We can see where all the transcripts are in 5 different tissues. And then we also started to play around with methylomes. This is the epigenetic code, and it's never been done in cannabis before. It's very hard to do in cannabis because cannabis, the genome is 63% AT. So there's 4 bases in most genomes, A, T, C, and G. And the human genome has roughly an equal amount of all 4 letters, making it a fairly easier genome to work with. But as the genomes skew themselves more in the direction of one nucleotide pair or the other, it's kind of like putting a jigsaw puzzle together where all the pieces are the same color. You end up having less entropy and signature in each of the pieces, and so you find yourself confused more often. Well, when you want to do methylation analysis, you have 2 choices. You can use some of these single molecule sequencers, but they're not good at doing it in plants because plants' methylation patterns are far more complicated than microbial ones. So you can hit microbial and maybe human methylation patterns with the single molecule sequencers today, but they don't handle plants very well yet. So we had to resort to a tool that's known as bisulfite sequencing. This is something that will convert the DNA if it's not methylated into it. All the cytosines in the genome, if they're not methylated, they get converted into a T, into a different base. And so by sequencing this before and after a conversion like this, you can figure out all the cytosine bases in the genome protected by the methylation mark. However, the genome goes into something like 88% AT. It becomes so AT rich that you can really, you get lost trying to put the reads back into the genome. You can only map half of the genome back with this tool. But it's the only thing we've got today to handle methylation. And so we ran that on 4 different tissues to get an understanding of where all the promoters are in the genome. The promoters are the regions in front of the genes that are usually responsible for turning the gene on or off.
Right.
And when they're methylated, they're silencing the gene. And when they're unmethylated or hypomethylated, they're turning the gene on. And so we've got beautiful pictures now of THC synthase and the adestin gene, which is involved in some seed development, those methylation patterns getting turned on and off DNA. One reason why we're excited about this is it does give you it gives you better paleogenomic tools. If you ever want to look at ancient cannabis samples, you can't do it with RNA. RNA is one of these molecules that breaks down really readily and very quickly.
Right.
It's that way on purpose because it needs to make those RNAs to send a signal and it needs to kill those programs afterwards. And so there's all this feedback mechanisms in cells to destroy RNA after it's been expressed. Methylation, however, sticks around for a really long time. You can actually pick up methylation signals on ancient DNA samples. And so it can give you some understanding of what genes are turned on and off across long time horizons. And so there's an interesting aspect to methylation data in that regard. But that information is now together and public, and people can kind of comb through it. That was leading to your final question, which is, what did we see in this data regarding pathogen response?
Right.
And on the pathogen response front, we took a little bit of a myopic approach in that we looked at all the genes that have been published in hops that play a role in pathogen response, particularly powdery mildew resistance, because powdery mildew is a type of pathogen that's very cheap to detect. There's not only PCR assays for it, you can visually see it. So you can tell if a plant is powdery mildew resistant or not, if it's surrounded by other plants that are loaded with it and it seems to not have it. That's not the case with a lot of other pathogens. You can't see them, so it's hard to phenotype them. But we looked at chitinases. Chitinases are enzymes that are throughout the kingdom, the plant kingdom. You see them in all plants, and you also even see them in fungi.
That's what I was about to say. Yeah.
Yeah. So that's an interesting game. Some fungi express these to kill other fungi. Trichoderma is one that makes chitinases to kill Aspergillus. So it's a weapon that's used to fight off fungi because the cell coats in fungi are made of chitin and another molecule known as a beta-glucan. Yep. And beta-glucan is something plants have evolved to fight as well with proteins known as thaumatin-like proteins. These things chew the other thing that's inside of a fungal cell membrane. So we looked at those 2 gene classes and then another one known as MLO, which is a mildew locus that was discovered, or mildew resistance locus that was discovered in hops and in other plants as well. But there's some work done in hops trying to map this out. So all in all, that was about 82 genes that we just went through and said, are there any large copy number changes in these genes that might correlate with pathogen response? And we're starting to find some of those patterns in Jamaican lion. There's a handful of thaumatin-like proteins that have copy number amplifications that seem to correlate with powdery mildew resistance. And there's some chitinases as well that are sticking out. So those have been interesting to look at. But I wouldn't say it's the final chapter on that story because I think we have found the genes responsible for powdery mildew resistance in Jamaican lion, but I don't think you can necessarily apply those to all the other plants out there because I think there's 5 to 10 different modes of resistance out there, and we just happen to be looking through the lens of one of them.
Right. No, that's a really important point when people read this data, that it's very fascinating and it gives you insight into this one mode of action. But like we were referring to earlier, the picture is almost certainly more complex, could be other things going on. So that's super super fascinating. There's a couple things that I wanted to ask you to define in case anybody's listening and has gotten a little confused. One is, can you briefly describe simply what methylation is to listeners, which normally my exposure to it is around like heavy metal resistance in plants and that sort of thing. But yeah, just briefly describe what that is, put that in context for listeners.
Yeah, so the ATCs and Gs in the genome faithfully replicate themselves in cell division, but there are other marks, almost environmental memories that get written onto the DNA, and that they get written onto the DNA in the form of methylation. They tend to methylate cytosines, Cs in particular. There is some methyl A that you see in bacteria, but the most common methylation pattern you see is a methylation signal that gets put on the cytosine. Now, there's a couple other forms of methylation on cytosine that I won't get into, but in neurons in humans, there's 5-hydroxymethylation, and there's a variety of other forms of methylation, but it's still all working on the cytosine base, just different methyl groups hanging off of it. But this is usually something that's done to make the DNA coil and to contract. It's when you add a methyl group, it makes it a more hydrophobic molecule. It's like adding more carbon to it makes it greasier, and so the DNA changes properties, and the methylation tends to also attract other histones and other proteins to it to wrap the DNA and coil it. And a lot of gene expression is based on just uncoiling the DNA, getting these histones to move out of the way, getting the DNA to relax so that an RNA polymerase can come in and start to express a gene. So you tend to find the regions of the genome that are getting silenced get heavily methylated. The cytosines get methylated. And then when they're getting expressed, they tend to erase that methylation pattern. Now, sometimes, in rare cases, this methylation is accidentally heritable. It gets moved through sperm and meiosis and into. They've shown in rats and mice that there can be methylation signals and small RNA molecules that transfer environmental information into the offspring. It's a very, very subtle effect. You need very large N numbers to really prove this, and it's probably a minority of the heritability that's going on in genetics. But it's fascinating. It's one of these. It's one of these things that's brought back the debate between, Darwin and Lamarck. This whole Darwinian genetics,
nature versus nurture.
Exactly. It's back, and it's good that it is back. It's been a long time. Lamarck was silenced for many years, but the reality is a little bit of Lamarck was right. And that there are aspects of your environment that will get written onto your DNA, and a small portion of that may in fact find its way into the offspring, such that the environment can actually change the genetic outcome in future generations. But again, it's a very subtle effect, and not nearly as powerful as the mutations you might see that are actually written faithfully into the DNA.
Yeah, the thing that pops up in my mind to conceptualize this in a totally different way. My wife is a mental health therapist. It reminds me of the concept of intergenerational trauma. It's like, these subtle things that can, get passed on that relate to how, somebody grew up and their environment and everything. But it's interesting, and I know we're deviating, but I think this is something in general I wanted to ask you about, misconceptions around genetics and genomics and stuff. So the concept of epigenetics is talked about a lot. It's a very, it's a big sort of buzzword in popular science, pop culture science and stuff like that. And so I one of the questions I was going to ask, and maybe just ask you now, what are some of the misconceptions that you hear kind of popularly shared about genetics, genomics, and particularly epigenetics, whether it be related to plants or humans, that you'd kind of like to set straight?
So the scientific field is really excited about methylation for the complete opposite reasons that pop culture likes to magnify its effect. The pop culture likes to almost guilt people into their actions today are going to hurt some child 3 generations from now, and that is
right.
Such remote garbage. It's hard to hang out on, but but we're excited about it because it teaches us about the machinery of expression and the cell. When we see the cell methylating certain things, it tells us, it gives us another vantage point on what's going on. It's one thing just to measure the RNA that's coming off of these, off the genome. It's entirely another thing to have and to know which parts of the genome it's methylating as a result of this, because that implies there's a feedback mechanism, that there is something else in the cell that is responding to a signal to make something transcribe, and maybe even a feedback mechanism to turn it off. And so we like having those circuits all spelled out. So we're really excited about epigenetics for that reason. But a lot of that often gets pushed into pop culture as being, your actions from 3 generations ago are now impacting the direction of the human population. And there's very, very, very thin evidence for those things. I don't doubt that some of those studies are in fact real, but the Ireland famine is one that comes to mind. But it's such a weak p-value and such a weak effect that you'd be better off worrying about whether you're staring at your phone too long walking into a street. I mean, it's
really.
There's stronger selective pressures out there that are likely to affect your genetics than some of those epigenetic effects. So it's unfortunate, but it happens. The science often gets interpreted politically, and I think epigenetics is one that has a sort of guilt associated with it that's being leveraged politically.
Well, yeah, and for people that are not scientists or have a very vague understanding of the research that's gone into this, it can really freak people out unnecessarily. It can cause a lot of stress, like you mentioned, that really isn't warranted. And another example that comes to mind is concern over different types of waves that are passing through your body, and the effects of gravitational waves and this sort of stuff that some people, when they start hearing about, like quantum gravity and quantum mechanics is a big one, where people start to hear these concepts and then they get really strung out worrying about these effects of things that, the effects may be real, but they're so small in comparison to other things that have much bigger pressures on you, like you're saying, that it's like it's not really worth exerting the mental energy to worry about that when you have much more real issues to deal with, that you probably don't have to worry about gravitational wave distortions being caused by comets and things moving through the galaxy.
Strung out is a great way to describe string theory.
Yeah, right. Yeah. So going back to the research study now. So, well, I guess one thing I want to summarize for listeners too, just to kind of talk about some of the techniques and the things you've described. So we talked about basic DNA sequencing, and that is looking at the possibilities essentially of the organism, what's there, the foundation to work from. Measuring the RNA allows you to understand what's actually at play within that DNA sequence. And then the methylation piece, being able to look at that, helps you understand how the environment is influencing these things that are at play. Is that a good, super simplified version of explaining that?
It is. And, methylation in many ways is I actually like the RNA data better because there's so much more information content there, and it's more accurately mapped. But the methylation is kind of another confirmation. When you see promoters have methylation patterns that promote the expression of the genes, you have more confidence in actually what's going on. So it's just another view onto the genome. But yeah, that's effectively the central dogma, right? As we have this genome, it is the hand that you're dealt, and if you don't have certain aces in that deck, you're never going to get them, no matter how much light or water you put in that plant. So if you don't have a THC synthase gene, you're not going to coach that plant into making THC some other way.
Yeah.
However, if you do have a THC synthase gene, the gene doesn't necessarily predict that you're going to make 7% versus 14% THC.
Right.
At least that gene doesn't. We're hunting down a couple other genes that are related to the precursors in the pathway that might in fact have a bigger impact on the magnitude of expression. But right now, the genetics between THC and CBD, they really just give you the ratio to expect. But they're not going to give you, if you push the magnitude of expression of a given ratio too far, it will break the USDA regulations. And the genetics today aren't going to predict that. I think in a couple of years, we'll know that. We'll know that there's a couple genes upstream that everyone's looking at that are involved, AA3 and AA1, and there's a lipotylic acid cyclase and kernel transfer. All of those things are great candidates for mutations that may in fact be driving the magnitude of expression between a 7% and a 14%. But if you have a 14% CBD line, you're going to be risking that popping hot. It's going to probably approach that 0.3% under the given regs that are there. And so a lot of people are unfortunately trying to find 7% lines, grow twice as much material, only so the material, 99% of it ends up going into an extractor where the ratio no longer matters. It's absolute insanity by regulation, but it's what's there. But to the point of your analogy on cards, if you're missing both aces, CBD and THC, you're going to be making the precursors, the kings, if you will. And that's so the genetics can be very, very predictive, and they can be predictive at a very early age, and they can also scale. That's one thing that I don't think everyone yet appreciates. While they probably are never going to give you and never going to be a replacement for an HPLC value, you're probably never going to have a field portable HPLC that can run thousands of samples a second. That will be doable with genetics. It's just a matter of time. But it's much harder to imagine getting that to work with an HPLC system that's field portable. So they're complementary, and we always encourage people not to view them as one versus the other, because there are many more things that you can measure with genomics, and its price points are changing so radically. You should pay attention to it, but they're always going to be complemented by people who have good analytical testing equipment.
Yeah, no, I think that's an excellent way to summarize that. It is all complementary, and even within analytical chemistry, there's sometimes a strange competition between analysts that prefer kind of traditional chromatography and those that like to rely on mass spectrometry. And you've got some people that are very resistant to, they kind of want to stick to chromatography, and that's what they do. And others like really trust spectrometry. And then, obviously, they blend together. You use mass spectrometry as exactly. But there is a funny, within the science world, there are these weird little camps and tribes, just like anywhere else, where
and they exist in sequencing too. We all bicker over what's the better platform to use.
Right. Yeah. Are there any other lessons that that you gleaned from this recent research project that you want to talk about? One thing we mentioned, but I don't know if we really got into it, was the relationship between these cannabinoid acid synthase genes and the pathogen resistance. We touched on it early on, but I don't know if we really got into it.
Yeah, so the ongoing debate in the field is what is responsible for residual THC expression in plants that don't have a THC synthase gene.
Yeah.
Why are hemp plants popping hot? And the number one culprit for that is CBD synthase. Whenever people clone that into yeast and run it, they tend to get like 20-1, 23-1 ratios of CBD THC. So the gene in yeast will make promiscuous THC. You can alter the pH of those conditions and even get those genes to make all the other cannabinoids. In fact, there's a really good presentation I'd recommend people look at. It's a CanMed presentation from this October from Andrew Horowitz from Demetrix. Demetrix is a synthetic biology company out of Jay Kiesling's laboratory. And Jay Kiesling is well known for cloning a lot of the plant genes into bacteria and into yeast. So this freaks a lot of people out, but there's actually really good science that you learn when you isolate a gene into a model organism like that, because you can perturb it in isolation of all the other cannabinoid genes that are in the cannabis genome. You take one of them out and put it into yeast, and then you can play with the pH and you can play with the temperature and all these other conditions, feed it different precursors and see what happens. And what they did is they fed those yeast hexanoic acid and they got THCP. So they actually found THCP before we found it in the plants. The Italians discovered it very recently, but if you actually go through the literature, you realize, no, they were making this in yeast, and they learned that the synthase genes in cannabis that we currently have, if you feed it octanoic acid, will make THCP. So we don't need to go be looking for a different THC gene. It's the same gene will fold THCV and THCP. We need to be looking upstream, maybe in the pathway that's governing what makes hexanoic acid and what makes octanoic acid. So we're beginning to be able to use synthetic biology to teach us about the plant. We did a very similar thing in this study, in that we took a couple of the genes that we saw were related to pathogen response, and we cloned them into E. coli. We took a thaumatin-like protein that was amplified in the Jamaican lion genome and also amplified in a couple samples that we got from Colorado Seeds and from Minibus, Mark Jordan's group. And those ones we put into E. coli, and once we had that cannabis gene getting synthesized in E. coli, we can make gobs of it and then apply it back to microbes that we know infect cannabis, like Fusarium and Aspergillus and Penicillium, and see if that peptide actually kills those microbes, at least in a petri dish. And we demonstrate some of this in the paper. It's not killing, we don't have evidence that the TLPs, the thymidine-like proteins, are killing Aspergillus, but we do see that it's retarding the growth of Penicillium species and of Fusarium oxysporum. The next experiment we have running is grabbing the other gene we found that was correlated with this resistance, which is a chitinase. We have that getting cloned and expressed in E. coli as well. And most of the plant literature shows that if you combine these two, you get a better 1 + 1 = 3 effect. And you really can knock down the microbes once you have both of them present. So we're in the process now of taking those cannabis genes and just making gobs of that protein so we can test out the theory. Are these the proteins that are responsible for keeping cannabis powdery? Are they some of the proteins, I should say, not the only ones, that are making cannabis powdery mildew resistant? And can we consider those as like a foliar spray? It's one thing to have the genetics and know how to breed for resistance with this particular form of resistance. That's very helpful, but it might take time. But there are some jurisdictions, particularly here in Massachusetts, where changing your genetics isn't easy. You have to kind of run with the genetics you put into the seed to cell tracking system. And if you want to add new genetics, you got to go through a lot of paperwork with the CCC, and it's a big hassle. So some people are kind of locked and loaded with the mothers they have, and they're looking for ways to fight off these pathogens with native peptides from the cannabis plant, not with some foreign molecule that's going to end up in a vape pen. And so we're really excited about that side of the market because we like the idea of enzymatic pesticides, because enzymes degrade.
Yeah, exactly.
And you can destroy them and you can get rid of them. So you can use them in a really ephemeral nature, and they're not going to end up like myclobutanol concentrating into a vape cartridge. And we'd rather do this with peptides that we know are from the cannabis plant because the consumers are already consuming those.
Exactly.
We don't want to grab something foreign that might trigger an allergic reaction. We want to grab something that has been time tested with human biology for thousands of years. And if it's come out of cannabis, it's probably been through that ringer before.
Yeah, that's super exciting, using these technologies to think about different types of pest management. And something that came into my mind then too is, we've been talking about pathogens, but what about genes related in some form or another to beneficial relationships that the plant would have with microorganisms?
So the thing that kind of shocked us that came out of the paper was if you look at the RNA expression in the plant, the most heavily expressed genes in the genome are these chitinases. However, and they're also the most differentially expressed genes, meaning that the different RNA, the RNA expression is quite divergent between tissues, but it's completely absent in the roots. And I think the same was similar with the TLP. So why is it not expressing pathogen response to the roots? Well, we think that's due to a lot of the work people have just demonstrated before us, but these mycorrhizal interactions you might have in the root systems, if you want to foster mycorrhizal interactions, you don't start off that handshake with a toxic chitinase.
Right, yeah. Come on in and let me kill you while you're at it.
Yeah, so that's kind of fascinating to us. The roots are not making this stuff, and that's probably to have some type of commensal relationship with the fungi that are in the soil. The other thing that was kind of interesting along those lines is that deletion that we see so commonly in the type 1 plants, which is a cannabichromene deletion. It's quite present in a lot of the hemp lines. And many of the hemp growers they're working in breeding programs to cross and capture that cannabichromene deletion into their hemp line so they can we think that's what's responsible for the THC levels that you see in type 4 plants. You'll see some type 4 plants out there that are like 100 to 1, and they don't have THC or CBD synthase. So the most likely place that residual THC is coming from is probably the cannabichromene cluster, and so people are looking to just try and get rid of that, particularly if it's already known to exist in the population and we can just do a couple crosses and get there. But in the process of doing that, we have to pay close attention to those other 4 genes that are on that deletion that are involved in pathogen response, because that may alter. You may breed yourself a really compliant hemp line that is loaded with botrytis and powdery mildew. And that's a little bit more frightening to us, actually, because the USDA has nothing in the regs for microbial detection.
No.
You can have it loaded with aspergillus, and as long as it's below 0.3, it's cool. And that's a bit of a bummer for those who are looking at CBD flower for like a smoking program. I mean, there's a lot of success in people swapping out CBD for tobacco. Or swapping tobacco out for CBD. I think Switzerland's been doing a lot of that work. And that's a much safer alternative, as Donald Tashkin's shown. But I don't know that I can say that if it's going to be hemp that can be loaded with pathogens. So we need to do more work there. That's still just a theory that these genes are missing when you select for cannabichromene deletions. There's a lot more work that needs to go into proving that those are in fact more pathogen susceptible. We haven't done that work. We just kind of pointed out the fact that it's a common deletion. People are probably going to breed for it, and it's going to come with these genes getting knocked out. But one of those 4 genes is a gene that's involved in viral response. And viruses, we know, we're now starting to be able to detect those with the RNA sequencing data that's been coming out of a lot of the laboratories. And the discovery phase of cannabis viruses is really just beginning. There's only a few that have been really quantitatively nailed, but I think we're going to see 10 more in the next 2 years. And that's going to change, how we manage those as well.
Yeah, I feel like I keep seeing, like every year since, I don't know, 1 or 2 years ago, I'm starting to see, or people are messaging me saying, have you heard of this virus that's been identified? And sometimes these viruses have very cryptic names because they don't have a name. It's I can't remember the name of one I came across.
Called cryptic cannabis virus.
Yeah,
1 and 2.
Yeah, it is. Yeah, exactly.
Those are in NCBI, and I can't. I don't know. We made assays for those. We've never found them in cannabis, and it's probably it's very rare. The challenge with the viruses is that they're usually RNA based. Sometimes they're viroids, and they don't even have a protein shell in them, so they're very susceptible to breakdown, which means it's unlikely they're going to make it to your laberatory intact. Across state lines, and very few people want to hang out for that experiment. That if they think they have a virus, they torch it. They don't. Let's put it over here so you can play with it. It's a bit to ask, saying, hey, can you save that one for me and ship it across state lines? So what usually means is you've got to teach people how to do RNA extractions in the field so that you can get something that's converted into DNA or is on ice and can survive the shipment to a laboratory. And we're just not there yet as a community. We're still getting some of those tools out to the masses so that people can do on-site RNA preps. But the ones that are tightly linked to a close university can probably get those studies done. And we're starting to see that. I think Dark Art Nursery did some work looking up that was a viroid, that was a hops latent viroid. But there was also some excellent work out of Israel looking at a lettuce sclerosis virus. They actually demonstrated white fly transmission in that paper. So the pest management is actually playing a role because some of these pests are actually the vectors for the disease. But we've built quantitative PCR assays for all 3 of those for cannabis cryptic virus, hops latent viroid, or hops latent viroid, and then lettuce sclerosis virus. And we're just in the earliest stages of working with people to get them to test these out. So if you know people out there that are interested in quantitative PCR tests to monitor that, we have the tools. We just don't have the grows or the gardens or the places to deploy them. We need to partner with people to see exactly how to put those things to use.
Yeah, I think it was the hops latent viroid that someone asked me about very recently, and I hadn't heard about that in the context of cannabis at all until they mentioned it. One thing that I was wondering is there any way to test a plant sort of generically to understand if it is likely to have a virus regardless of identifying what that virus is? Because I know a lot of cultivators battle this, sometimes viruses appear as like nutrient deficiencies. They have very similar symptoms. And I have cultivators that ask me a lot, how do I quickly know whether this is a nutrient problem or a viral problem?
So this is actually an industry-wide problem that's not unique to cannabis. Viruses, they don't have necessarily conserved sequences that we can rely on to universally capture them and sequence them. So if you look at fungi and bacteria, there's tricks that you can play. They all have to have ribosomal DNA operons, right? So you can design primers that will amplify all yeast and mold. You can design primers, or I shouldn't say all, most, because there is even some divergence amongst primer sites you might pick to try and amplify all of these things. But Likewise, with bacteria, you usually go after the 16S region, and that a single set of primers can probably get you everything in the bacterial side of things. We don't have the analogous primer sequences you might target for viruses that would be conserved across all of them. They're having this problem with coronaviruses right now, that they have to engineer new assays for these viruses so they can spread them around the world, and the virus mutates a little bit, so they have to have multiple different primer sites that target the coronavirus. So what people do in the case where they can't use something as fast as qPCR is they resort to RNA sequencing, which just captures all of the RNA that's present, sequences that. Sometimes they'll try and deplete the really common ribosomal sequences that are in the RNA, so they get more efficient sequencing, but it's another expensive step. And then they blast that all through a DNA sequencer, and then filter that information for sequences that look like they're from a virus. So there's several viral databases out there that you can leverage that will scan your RNA sequencing data for virus content. But that's expensive. That's you're talking about hundreds to thousands of dollars to do studies like that. So it's not something that you just do on a whim. And so you find yourself in as the scenario of doing lots of tests for individual viruses of suspect. Now, I've not seen tobacco mosaic virus ever show up genetically. I've seen people say they have it due to some pictures, but I've not seen it actually show up in a test that proves it was that. It's probably something very related, but not exactly tobacco mosaic virus. It's probably a hops mosaic virus or something. Yeah. There's a few of those that have been listed. So it does make for a bit more cumbersome of a test. How they handle this in human clinical studies is they tend to have multiplex tests that look for a panel of rhinoviruses. So they first scan 20 or 30 of them with qPCR, and then depending on what hits they get, they might do narrow it down to some other ones. So that's at least what they're doing with the coronavirus. They have a test that looks at a handful of common cold viruses, rhinoviruses, and norotype viruses, and then if they see it's negative for all of those, they've graduated to maybe a SARS or a coronavirus test. So we'll probably see something similar like that in cannabis, as there'll be a two-step screen. And if you really need to know because it's plaguing your entire grow, you might graduate to doing DNA sequencing and RNA sequencing to thoroughly figure out this is something novel that's never been seen before, and you want to crush it. That's usually what you do.
Yeah, fascinating. I know that's probably I know growers want that easy sort of detection fix, and I'm sure it's disappointing to hear that it's just not there. But I know many growers that have tried to battle what they thought were nutrient deficiencies for far too long, trying to battle what actually ended up being a virus, and just wasted a lot of time and everything. And my response is usually like, if you're really worried about it, can you just torch that plant and move on? Rather than wasting so much energy and time. And some growers are now getting to that point because their fields are so big now, they're not worried about saving a single plant or a handful of plants. So they're able to just cull the ones that seem to be causing problems and move on, which would be more typical in bigger forms of agriculture. Changing gears quite a bit. What are your thoughts on how genomics research is going to influence cannabis taxonomy in the future? Because that's a really muddy area in general. Cannabis taxonomy is one of these things that nobody, even, botanists and chemists and geneticists, it's hard to get on the same page on how to classify plants or even how to define what a species is. But where do you see that moving towards in the future?
So we're in this weird time frame where the entire phylogenetic tree of life is getting redefined. And so I'm kind of waiting for that dust to settle before I weigh in, because it's they're starting to do it genetically, which is if you're 95% identical to this other microbe, you're the same genre or the same species. They're trying to make metrics like that to begin calling these, and it is needed because we have a lot of redundant nomenclature even in the microbial side of things. If you try and get Aspergillus species out of ATCC or other cell banks, you'll find that a lot of them haven't been sequenced before, and then you apply primers to them from the literature that are supposed to hit them, and they don't, and then you sequence its genome and realize it's not what they told you it was. So a lot of this stuff has all been done with eyeballs, people looking at the morphology and looking at these things on plates. And of course, organisms can express really bizarre phenotypes. I mean, the classic case is like a caterpillar and a butterfly, right? Same genome, complete. You wouldn't, if you looked at those things, you wouldn't call them the same species.
Right.
If you look at Aspergillus species, they can be just as genetically diverse, yet they look identical. So the look of the species is no longer trustworthy for classifying them. Now, speciation in general usually involves whether they can continue to cross. Right? We have cannabis plants that all cross with one another, so they're arguably the same species. Now, if you want to subdivide them into subspecies, indica, sativa, and all that, that's great. But we probably need some paleogenomics to really nail that. If that's in fact the story, that the indicas came out of one geographic region at one point in time, and the sativas were more equatorial, then maybe that will bear itself out. But right now, they've been so mixed inappropriately named during that mixing process that the sativa indica thing doesn't match up to any of the genetics right now. So I usually don't get involved in a lot of debates on whether we're going to call these things chemovars, cultivars, or strains. I tend to mock them actually, because I don't find it a very productive conversation when the whole tree is getting rewritten. We can argue over the taxonomy stuff later. I tend to just read the DNA, because that's the answer you're really looking for. What you're looking for in this categorization is, how do these things differ biochemically? And so chemovar is a great name, but it doesn't, right now, the chemovars we're measuring aren't really measuring the flavonoids. So I don't view that as a legitimate way to necessarily draw the taxonomy. But ultimately, it's the heritable units that govern the taxonomy. Taxonomy is DNA, and anyone who tries to tell you otherwise is going to be proven horribly wrong in time.
Yeah, no, I think that's really important to point out. I remember when in some of my early graduate work studying mycology, how frustrating this issue was of learning the taxonomy of, different fungi, and then tomorrow it changes, because just like you said, there'd be some genetic work that's done and they realize, oh, these things that look identical even under microscopy and everything, they seem the same, the spores look the same, have same ornamentation, but they're different genetically, and now we've got to call them something else. So students, everything you learned, wipe your brains. Now we're changing the whole system, and even botany too. I was a botanist with the BLM for Wild Bureau of Land Management, scouting native plants around Oregon and everything. And I realized it had been a while since I've gone back and looked at, the scientific names of a lot of the plants that I worked with. And so just a few months ago, I went and looked and saw that about half of them that I was studying that had already gone through like genera changes and that sort of things, have already gone through changes again and been reclassified and split and everything. And so it's just a headache of trying to keep up with all this stuff.
To give you a sense of the wave of headache that's coming even further, the last study I heard, I think was in the UK, they're going to sequence 1 million species. All right.
Yeah.
So once that happens, everything that you've learned, you got to rewrite. We see the Aspergillus species are getting renamed all the time. Braziliensis got carved out of Niger. So I have no doubt it's going to happen in this field. But I guess I've been advising people in cannabis, just stay tuned and be patient, because this is the wrong time, I think, to fight over those terms. Because we're just in the earliest stages of sequencing everything. And once we have a map of like 100,000 different cannabis genomes, I think we'll have a picture. And we probably aren't going to try to force that picture into sativa, indica antiquity. We're probably going to say, okay, the autoflowers are over here, and the genotypes that have these types of clusters are over here. And it'll be named in a coordinate system that's probably more related to large segmental duplications or deletions that are related to its diaspora around the planet.
Yeah, I agree. And I think the main thing, any listeners that are banging their heads against the wall about how to talk about cannabis varieties, the main thing is to think about, like, what are you trying to communicate in the first place, and what do you care about? And that can sort of drive your vocabulary a little bit. If you're if you're looking at cannabis varieties and you're trying to understand chemical differences, then talking about chemotypes and stuff could be valuable as long as you understand that that's a tool for us to communicate things, not necessarily some hard-coded taxonomical
Religion.
structure. Yeah,
Yes.
exactly.
Yeah. And I think that's important in science in general, because it's very rare that science drives to a consensus. We want it to.
Yes, yeah.
But
Yep.
I mean, I think they changed the definition of the kilogram last year, right?
They did. They did. Yep.
Yeah, so you're like, oh shit, that can change.
Right.
So I thought that was like tied to some astronomical unit of the wavelength of light or something. I didn't realize it was ever going to change, but they do. And consensus. Hey, there was a consensus against marijuana for 80 years. It was pretty successful.
Right.
And clearly that's in the latest stages of changing today. So, yeah, I think you're right. It's about communication, and sometimes the communication that's most popular is probably technically wrong. But if your goal is to communicate, think the most amount of people that will understand what you're saying. Sometimes you just use the language that conveys that most simply. I think that's probably the case with what's happening with strain. No one likes that name because it's a microbial term. But if you do a Google Trends on strain versus cultivar or chemovar, there's no one talking about cultivar or chemovar. Everyone's continuing to talk about strain. So if you want to effectively communicate to large audiences, sometimes you have to use those terms. The reason people put things in Latin is because it's a dead language and the language doesn't change.
Yeah.
And so we don't yet have chemovar or cultivar necessarily in Latin. I think cultigen might be technically a combination of an acronym of Latin terms, but I think it's all about making sure we have communication and not necessarily hard fixed religion, because if there's one thing we're going to learn in the next few years is that whatever we're saying in this podcast today will probably be partially mocked as being wrong in a few years.
Yeah, I mean, there's two things that come to mind there. I mean, one is I've always been a fan of Richard Feynman and his work, particularly like as a science educator.
Yeah, I love that guy.
Yeah, he did a lot of great work for science education in general, much less, the physics side of stuff he worked on. But something he talked about in one of his late books called The Meaning of It All. And he talks about the problem with science education in our culture is that most lay people expect that's what science does is it produces facts. And it produces, and those facts, everyone can form a consensus around them because they're facts. They are known. They're hard, objective truths. But the reality for people that are, actively doing research and, diving into all this stuff is you realize there aren't a lot of hard facts, if any, and consensus is harder than you would expect, depending on what you're trying to get consensus over. And what Richard Feynman said is that ultimately what science does is it gets at answers to questions. It may not necessarily provide you the hard answer, but it gravitates towards the answer, and it's always moving towards that answer, even if it doesn't quite get to that very specific hard truth. But when you talk to people, like for instance, on a political stage, and you're talking about, let's say, climate change, people want to know. People want to know the facts. They don't want to know that you've got some general idea of where the science is moving towards, and they don't want to hear about probabilities and confidence intervals. They just want to know what's the state of things and what do we need to do. And so when a politician is willing to just assert solid, bold, flat claims, people resonate with that because it's a lot easier to digest than the actual muddiness of science.
Yeah, no, it's very true. It's very true. It gets weaponized oftentimes.
It does.
My view on this is they only separated the church and the state so that they could replace the church with people in white robes versus black robes. And now the scientists are propped up as some kind of priest that can read the code of life.
Yeah.
And everything that comes out of their mouth is ironclad truth, and we're going to drive politics off of that. And that's nothing could be further from the truth. What really is happening in science is we're generating questions, not answers, most of the time.
Yeah, exactly. No, that's a great point. It is generally about trying to figure out what are the better questions to ask, and then that drives what research you're going to do and how you're going to approach a question. And Isaac Asimov, another writer, science educator, even though he wrote a lot of fiction, he had a paper that he wrote in response to someone criticizing a statement he made about basically kind of like what you said just earlier, that, in 10 years, everything we're saying now, a large part of it is going to be, mocked at and thought of as wrong. He made a statement like that, and someone said, well, if that's the case, then, does that mean that essentially a lot of things are wasted and that anything could be overturned at any time? And he was like, well, no. Like, what that means is just that our understanding gets sharper over time, and it feeds off of what came before. It's not that what's going to come after is going to be totally, irrelevant and in total opposition to the science that's happening now. It's just going to keep pressing forward. And I think the example he used was like the spherical nature of the Earth, and how, eventually we learned that it's not a perfect sphere, and that it's, sort of like a lemon in a way. It has, this sort of oblong nature to it, and it even kind of like wiggles and moves. And before that, it was assumed that, the Earth was flat.
It's not flat?
You're right. Yeah. All these things. And so just pointing that, these aren't total overhauls of what came before, but they're refinements. And, it's like getting the clarity tuned up even more as things go on. I think that's important for people to understand, that, what we're saying is not that all of this is just getting thrown out, and that, a lot of scientists are essentially wasting their time, but that, we're all standing on the shoulders of everyone that's come before. And people 10 years from now are just going to have a very different set of eyes than we have now.
Yeah, I mean, to me, that's probably the most compelling argument I've heard in this climate debate, is that the number of ideas that are human generated tend to be cumulative. Like there's idea bridging. And so the fact that we had twice the amount of population back when Einstein was around, or Edison, or Pasteur, right? If you did population control because you were concerned that we weren't going to be smart enough to figure out how to use things more resourcefully, you would have picked off, Einstein, or maybe Pasteur. And then where would the world be if we only had electricity and not the germ theory of disease, right? The fact that we have that we had the population we had back then, we are so much more richer and more resourceful today than we would have been if we actually choked the population back then. So I think it's an important, it goes back to the concept of humans not necessarily being a cancer cell on the planet. We're actually part of this thing. We have to learn as much as we can about it so that we don't unwisely use the resources that we have. But ingenuity is very important. And I think the evidence is pretty clear. If you look at people's standard of living, and if you look at people's income, they tend to become more environmental as that goes up over time. A lot of the environmental damage is done due to poverty, and more people are moving out of poverty than ever before. And so we actually have a really bright future that you won't hear about in the political sphere, because that's about selling fear that we're all going to die tomorrow from coronavirus and tsunami or something. But I think I'm very optimistic about the fact that the population is going to be fine, that I think science is going to be, it's exponentially building upon itself. And we often underestimate that fact. I mean, just now we're in the first generation of people who have access to all information knowable on their phone. Right? Like, school is now irrelevant. No one wants to say that, but it is, right? If you like to read and you're curious, you don't need school, you need your phone. And no parent wants to say that to their kid, but we didn't have that when we went to school. We had to go to centralized campuses where all the information was literally stacked on paper into libraries, so that in microfiche, so we could comb through this stuff and be near the people who are smart and who already read it. But now we can be near those people on conversations like this, and and we have all the information to That's going to radically change idea generation, and it's going to radically change people's access to capital and the ability for people to grow and to invent and to become more resourceful with the limited amount of resources we have. And I think it's really exciting that cannabis is like emerging from prohibition during all of this, because we now have probably one of the most resourceful plants on the planet, where people have been restricted from being able to really adequately study it. And now the gates are open, and we can study one of the best photosynthetic organisms we have that can literally feed the world with this thing. So there's a tremendous amount of potential, I think, for solving a lot of these climate concerns people have. I think just by doing what we do so well with hemp, Learn this thing, figure out its code, figure out how to tweak it, figure out how it interacts best with the environment. And that's going to be a combination of not its genome, but also the microbes that are in the soil.
Yep.
And the more we can learn about that, we're probably not going to come to a consensus about what it all means. But as Feynman's saying, we're going to be pushing ourselves in the arc of being more and more efficient with what limited stuff we have here.
Yeah, man. Yeah, that's such a great summary to bring this whole conversation back around. I really appreciate how much time you've been willing to spend with me. We've been going for almost an hour and 45 minutes. I have a couple of closing questions for you to cap us off. One is, what other research interests do you have beyond cannabis? I usually like to ask every scientist I interview that question.
So I'm really interested in other pathways that are in its neighboring plants lately. So I've been looking a lot at the and also this concept of xenohormesis that your previous guest, Kevin Spellman, will probably have a lot to say about it. But hops has a similar pathway that makes terpenes, and it makes a different set of compounds that I would probably call the analogous medicinal compounds in hops are the xanthohumol.
Okay, yeah.
Right? And those compounds are starting to show anti-cancer properties, much like the cannabinoids. We don't know really what receptors they hit and why, but everything that's needed to make those compounds has some homology to the cannabis genome. So it's possible the cannabis genome could be making some of those compounds as well. I don't know if it's going to need to be crossed into it or how we genetically engineer it, but it's something that it makes you turn back and look at all these other plants that we've taken for granted and start to ask, well, what are the secondary metabolites in those plants? Because the only reason we've paid so much attention to cannabis is that one of them has a really easy bioassay to tell you when you're getting something.
Right.
It's THC.
Yep.
We don't have that in hops. We might have beta-caryophyllene, which is a CB2 agonist, but you probably can't really feel that unless you're in pain and you take a lot of it. So all these other compounds that we can't really perceptually feel, we need to go back and look at because they've been the FDA system is selecting against these things.
Yeah,
and in many ways, the regulatory environment we have here in the United States favors largely capitalized companies that can then pay politicians to limit the regulatory space of the operators, and so the genetics tend to go through bottlenecks in those environments, and we end up having massive genetic shifts because of regulations. And so I'm somewhat fascinated about the impact of regulation on genetic drift because I think that there's genocides going on all over the place as a result of this. This 0.3% thing is going to create a massive, just like prohibition, create an enrichment for type 1 genetics and probably a depletion of a lot of type 3s. This 0.3 thing is going to have the opposite effect. It's going to end up meaning we're going to have to select a very narrow set of cultivars to move forward that are compliant with the USDA, and it's going to wipe out all types of diversity we don't know yet because those selections arguably are going to wipe out these other secondary metabolites no one's measuring.
Right.
No one's going to be measuring the canflavin pathways in the process of doing these selections. I mean, we have sequencing that looks at that pathway, but very few people are necessarily going to jump on that train right away. So the process of even putting in microbial regulations in place, this means plants that like and enjoy a particular microbial background are going to get eliminated from the population.
Yeah.
Right. Let's say you have the plant that needs trichoderma. Trichoderma is not harmful. Yet some of the regulations in some states don't have CFU language, right? Colony forming units are they don't know what the hell they're counting. They're just saying bugs, bad, right? It's really ignorant to think of microbiomes that way. So when you put that in place, all of the plants that can get through regulation then go through a genetic shift, right?
Yeah.
You get this selection for plants that tolerate low microbe environments. Well, those may not necessarily be the most productive plants from a secondary metabolite standpoint. No one's looking at that. They're just looking at maybe THC and CBD, but all these other flavonoids might be abolished in the practice of doing that. So I'm kind of curious at seeing where that's going. But the xanthohumol thing and the xenohormesis is just this concept that these plants are making these secondary metabolites to communicate to the things that propagate their genetics, right? So if you were to actually look at the number of cannabis cells and the number of genomes that are dividing throughout space and time, there's probably a good argument that it's winning that race compared to humans. that we are propagating more cannabis DNA right now than human DNA.
Yeah.
Because of the nature of how big some of these plants can get, how big their genome is, the fact that they replicate every 12 weeks, and humans don't replicate that quickly, means that some numbers I've drawn out suggest that the cannabis genome is actually done a remarkable job finding an ideal pollinator that likes to propagate it all over the world. And how did it do that? It did that by being able to make secondary metabolites that tap into our biochemistry to tell us that changes in the environment are coming or what have you. So this work is all done by David Sinclair. He's the guy who does a lot of work on the fountain of youth and resveratrol and many of these compounds that are involved in that pathway. But he's got a great paper out there called Xenohermesis, and it talks about how these plants, when the environment changes, they shift the secondary metabolites they make so that the things that eat them, their mTOR pathway gets triggered so that they start going into caloric restriction because the plant's biochemistry has changed to know that the environment is going to change and it's going to have an impact on the amount of material that it can feed the thing that's supposed to propagate its genetics. So that whole interaction space, I think, is the key to cancer. And if we can understand how those plants are signaling the metabolism of the things that propagate their seeds, we will understand cancer in a much better way, and a much more thorough understanding of cancer. Because usually cancer is a scenario of your own genome having a metabolic defect going on in the subpopulation of the cells, and that metabolism is getting triggered, and it's usually because of an acidic or a different environment, a mutation that's happened. And the things that seem to be having the most potent effect on our cellular metabolism are the vegetables and the plants and the foods that we eat. And that's what we need to better understand to really understand cancer. So I kind of like that intersection of plant metabolites and cancer. It kind of touches home for us. And it's a field that I think our myopic focus at the FDA is constantly deteriorating because they have this reductionist approach of looking at single compounds as opposed to symphonies of them. And we're at a stage now where we have the tools to look at symphonies. They just don't fit into their trials very well.
Exactly.
And we need to rethink whether their trials are the right way to do medicine. I happen to think they're not. That you can get a hell of a lot more power out of a study with 100,000 to 1 million people connected via observational trials on digital equipment like we do with peer-to-peer networks, and 5,000 or 10,000 people in a trial that is a subsampling of statistics to the point where you end up with Vioxx-like compounds that escape that process.
Yeah, I mean, and not to mention the way that the clinical trial system is set up right now, it creates such a high barrier to entry to even try to get through that process, Yeah,
it literally kills people, and there's no one willing to look at the seen and the unseen here. It's like, okay, the fact that a drug takes $500 million and 10 years to get through. Has anyone done a survey on the number of people that died waiting for that drug, knowing that it's available in Europe and it's available in Australia? Other countries have deemed it safe, but not here. And those people don't have the money to travel there to do it. So they sit here, they wait, and they die. And there's people who study this, and it's a large number of people every year. The Independent Institute does a lot of work on this called, and they're FDAreview.org. It's worth reading because they go through all of the scenarios where drugs that were approved in other countries, so they pass at least other countries' criteria, and eventually passed here, but 3 or 4 years later, and they count the number of deaths that occur because of this. And no one's responsible for this.
No.
And you can't vote them in or out. And I think they regulate now like 25 cents of every dollar. So this is something that is, I'm totally terrified that it's going to come into the cannabis space and somehow make the same mess of this space as it did the opiates. That being said, there's a lot of good scientists that work there that have all the right intentions, and they are trying to put genetics into all their programs. They're trying to build blockchains to track all the food. So they've got all the right vision. I just think they don't recognize that it's too big of a problem for one centralized organization to solve. And it's too prone to regulatory capture and corruption. That they shouldn't set up that honeypot. They should somehow decentralize it and have competing agencies that can perhaps invent different ways of regulating. Because it's certainly an important feature of a marketplace to have regulation, but not necessarily in the way that it's getting done today.
Yeah, and I mean, I also find it kind of funny that, a lot of times drug approvals get so hung up on needing to prove a certain level of safety, sometimes to kind of an absurd extent. I mean, right now we're seeing this with CBD right now that, with
So the liver tox stuff.
yeah, exactly. And, and not acknowledging the nuance around, some of that research and everything. But what's interesting is that the actual efficacy of a lot of pharmaceuticals that actually make it through the process are not that effective. Maybe for 20, 30% of the target population, they may be effective.
Right. Yeah.
And they have terrible safety profiles. And so it's, I don't know. It's a weird dynamic that the safety profile of a lot of these natural products that people want to research and try to understand how they can be elevated out of the realm of sort of, this alternative medicine sphere and try to understand like what they're actually doing and how to actually, legitimize them as medicines. It's very challenging, even though the safety profiles of a lot of these things are pretty well understood by this point.
Yeah, and I think the other big hangup they've got is there is this gold standard, almost religious consensus, as you were mentioning before, on double-blind placebo-controlled trials being the only path to ensuring safety of medicine. And I think that's being fundamentally challenged by, if you look at the work on what governs the placebo effect, it starts pointing back to the endocannabinoid system.
Yeah.
And so there's actually papers out there on the endocannabinoid system being involved in the placebo effect and analgesia, right? So how do you put cannabinoid through a double-blind placebo-controlled trial?
Exactly, yeah.
If the placebo effect we think is people's reactions, their ECS, their reduction in anxiety is caused probably by anandamide or something, that when they take something, they're taking control of their problem. They're taking action, and that action gives them confidence, and that boosts their anandamide levels maybe.
Right.
And so the very pathway that you're trying to interact with is what's involved in the placebo process. So I think that's just all going to create noise, and we should be looking at very large observational studies that we can enable now with these digital tools that we have. And that's probably going to give us a far faster and more, back up and just look at the vape crisis, right?
Right. Yeah.
So this is a COX-2 inhibitor, CBD. A lot of people are vaping CBD, sometimes THC, and they both have analgesic effects. And so I throw them into that category of Advil and ibuprofen replacements, right? Maybe even Tylenol. Tylenol is, in fact, an endocannabinoid mimetic, which makes AM404 with a shitty liver metabolite. But so people are vaping these things probably because they like getting a little relief from some pain and some fun. That took the field probably 10 months to resolve, right? Because everyone was sharing information. Rumor came out of this company's making crap. This one's got bad vitamin acetate. Ideas are flying. And with all of the networking we have, there's a crisis that was affecting maybe 1,000 to 2,000 people.
Right.
There was 9 million vape pens sold a month, 90 million, maybe 100 million over the course of the year. And we only had an incident rate of 1 in 45,000, effectively. Now, that's one piece of arithmetic that's important for how frequently we sample for these contaminants in cannabis. We're not sampling nearly deep enough to find a frequency that low. So even if we were testing for vitamin E acetate, I'd argue we still would have missed it, because the frequency of this creating a problem is so low that you need to up the sampling rate you do in the industry to find it. But that being said, can roll it back to Vioxx, right? Vioxx went through, probably killed 50,000 people, and it took the FDA 5 years to finally admit they were wrong, right? And why is that? Well, because admitting they're wrong after they were paid a lot of money by the pharmaceutical industry to put that drug through is harder to reverse, right? It's more crow to eat than you want. And many people don't realize the FDA right now is governed by the PDUFA Act of 1992, which means about half to 2/3 of the revenue going into the FDA comes directly from the companies that they regulate, right? This is a way of defraying. System. Yes, it's a euphemism for bribery. It's effectively, how can that agency with a straight face claim that they are out for the consumer when they're on the payroll of the people they're regulating? This is going to lead to really bad decision making, even if it has the right intentions. And the incentives aren't there. When they make a mistake, they're going to get a bigger budget.
Yeah.
So this to me is like, we don't need this near cannabis. The cannabis hasn't killed anybody. So what is the FDA? What they've already done to cannabis, if you just look at Epidiolex, has made it 30 times more expensive. Yeah. Made it an isolate, put in a strawberry flavoring that is registered as a pesticide by the EPA, and then added in like sucralose, which like gives people seizures.
Yeah,
They made it into a worse drug and GW has to kind of unfortunately put up with this.
Right.
and GW is filled with people who know
yeah.
that know the extracts are more effective, but they can't get something that complicated through. I mean, they got Sativex through, but they haven't gotten it through
Not in the
our
US.
system.
Yeah.
And
And
they
if
had
it
to go
does,
for isolate CBD.
right. Yeah. I mean, Ethan Rousseau and I talked about this in our interview and a little bit off interview too. But, the fact that they're struggling so much with Sativex, and that if Sativex does make it through the U.S. system, it might end up being a very different pharmaceutical altogether than what you would find in the U.K. or in Spain or things in Canada too. And it is really interesting, and I agree. I mean, I've been following GW for pretty much since I knew about them in, I don't know, long time ago in the early 2000s or something. And I've followed the work of several of the different scientists that have come and gone that have worked with GW, and I agree. I mean, it seems like, as far as I can tell, they understand the best way to actually apply these medicines. Some of the research they've done has actually highlighted, that these
They've
that
got
these
some of the best
extracts,
data out there.
yeah,
Yeah.
but they're having to play the same regulatory games as anyone else, and it's so common, especially in the cannabis industry, for people to bash anybody that is perceived as a dastardly pharmaceutical company. And GW,
Hey, they write the rules. They got to play by them.
right?
So I don't blame. Sometimes they write the rules when they're really big. I don't think GW's at that scale.
No, they're not, and that's something I try to remind people of. Is one, they were doing this work in other countries initially, where they were able to do things that they can't really do here, and some of the products. I mean, Sativex in particularly is actually a very impressive product when you think about it. It's not. I think there's a misconception that it's isolated THC and CBD, but it's not. It's a standardized. It's a standardized extract. Yeah, and Ethan and I were talking about
they do actually a really good job measuring. They're measuring things that the labs are measuring.
Right.
I think they're measuring sterols, a host of these other compounds that we're not seeing in the dispensary. So they have, with hands down, the best manufacturing process for this that there is. I don't doubt that one bit.
Yeah.
But they're getting asked to point that amazing scientific engine they have in the wrong direction.
Right. But tell us, what's the active?
Yes. Yeah. They're all active.
Right. Yeah. So it'll be interesting. I mean, I would love to see in the United States some process for getting herbal medicines approved as, legitimate standardized medicines. And, now we have what are they called? Like medical foods that you can get prescriptions for that often are usually just imported from other countries. But the example I always use is like standardized ginkgo biloba extract that you can get a prescription for, and it has standardized amounts of, certain chemical constituents. But it didn't go through it wasn't produced in the US, and it's an interesting process to even get it to be allowed to be used the way it is in the United States. And it's just one of those things that I think a lot of people don't realize, that there are alternative ways of determining what is a medicine and how to give people access. And that a lot of countries do have some pretty decent ways of handling herbal medicines. And this isn't a complicated problem to figure out. It's just a bureaucratic and political problem to figure out in this country.
It is. And unfortunately, I think it's that same process of politics and bureaucracy that are forcing these really odd distinctions between medicine and food.
Yeah,
food is medicine. And
it's all chemistry.
This concept of like recreational versus medicinal to me is like mind-boggling that people don't recognize that if you do the recreation, you won't need the medicine. It's
right.
They're one and the same. It's just when you get into a chronic state of disequilibrium, you might call it medicine.
Right.
But
yeah.
But that distinction is purely political. And it comes, I don't know where it comes from. It comes from some hatred of people getting high. And yet they happily to have, Bud Light commercials all over the Super Bowl. So.
Right. Yeah. I don't know. It's a strange state of affairs. Interesting time and place in history that we find ourselves. Hopefully it evolves in a more favorable direction in the future. Now we've already gone over 2 hours, so I'm beyond ecstatic that you've been willing to talk to me for this long. I want to give you a final platform here. Let people know how to learn more about medicinal genomics. Also, one thing we didn't even talk about, but is Cannapedia. So where people can actually explore some of the genetic data and even some chemotype data that medicinal genomics and other groups have been bringing together. So I'll just kind of give you a platform here to let any of our listeners know anything that we haven't covered and where they can find out more about your work and everything else.
Yeah, so medicinal genomics, we make, in addition to doing sequencing for folks in the industry, we make a lot of tests to look for different genetics in the plant. So THC and CBD and XY testing, we make assays for testing those markers. We have markers that look for fusarium and powdery mildew, and basically all the microbes that might harm the plant, and also all the regulated microbes. So we sell a lot of quantitative PCR kits into many of the labs that measure Aspergillus and Salmonella and E. coli and those things. So that you can find at Medicinal Genomics. The sequencing work, there's a lot of it put public on a site called Cannapedia. A lot of people don't know that we run that site because of the way it looks, but that's a medicinal genomics site for hosting a lot of this sequencing data. It's serving some purposes for research and also for IP. There is a blockchain component of that will etch the timestamps that your data is generated into a ledger that we can't control. So if we ever become evil on you, you still have your data recorded somewhere else. And that's all managed at the Cannapedia site. And then thirdly, we do run a scientific conference every year called CanMed. So if you're interested in more of these topics, there'll be many people presenting there. Last year we had Raphael Mechoulam, and he's come most years. I think this year might be a little tight for him because he's getting to the age where it's hard to make that travel from Israel. But other folks of his caliber, we try and bring in and have scientific discussion around crop yield. How do you breed for better crop yield? How are we going to handle appellations is one thing that's coming up that we think is really important for the cannabis field, so we don't turn into a monocrop in the U.S.
Yeah.
And then we're also hitting analytical chemistry labs and genomics topics. So submit your abstracts, take a look at it. It's usually, this year it's going to be in Pasadena, down at the Pasadena Convention Center. I think it's at the end of September.
Nice.
But it's usually around 800 to 1,000 people.
Oh, nice. That's awesome. I'll have to keep my eye on that, see if I can make my way down there. I've never been, but I'm not a huge, I don't know. I tell people I'm kind of a hermit. Doing this podcast and starting to finally get out in the world has been a big challenge for me. I've got to get out of my cave more. But trying to make it to more conferences regularly, and CanMed's one that I've had my eye on a lot just because of the caliber of people that usually show up there and the sort of higher level of discussions. It's one I've wanted to make it to, so I'll have to try to get down there. Thank you so much, Kevin, for coming on and chatting about so many topics. I've really enjoyed it. Keep me updated on any big moves that you guys make, or any new publications like this preprint that you shared
Absolutely.
with me, anything like that. And if anything big comes up and you want to come back on the podcast to talk about anything specific, just let me know. I'll be happy to have you again.
Absolutely, Jason. Thank you for the time, and keep up the great work. I love hearing these podcasts. I was running out of things to listen to. Now yours is on.
Well, I view that as a great compliment. Thank you so much.
Yeah.
All right, everybody.
Great.
Thanks so much for tuning in and listening once again. If you want to learn more curious about cannabis, you can go to cacpodcast.com, or you can find us on social media. We're on Instagram, Facebook, and Twitter. And we also have a YouTube channel now. And when we're able to get videos of interviews, we get those on there. And I've also started doing some site reviews and book reviews and that sort of thing, or at least editing some of the video that'll come up pretty soon. So check all of that out. And as always, please tune in next time. I really appreciate it. Thanks so much. Stay curious and take it easy.